Hey everyone, my name is Jesse Wiskovich and this is Group Advanced Concepts, Presentation 8. So for this presentation, we're going to focus on how we process and form nucleotides and ribose and how they're made and how we can also form ATP used for DNA replication. Then we're also going to focus on ATP production, and we can only use starting products of glucose and CO2. So there's two main ways we can do this. So the first one's going to be cellular respiration, where we focus on pages 7, then we go to 12 for citric acid cycle, and we go over to 11 for electron transport chain, and then 6 for ATP synthase, where we get a lot of ATP produced. The second process, we're going to focus on ribose and nucleotide synthesis, where we're going to also start at page 7 for glycolysis. We're going to move over to page 1 for pentose phosphate pathway. Then we're going to go to page 4 where we focus on PRPP, and then to page 5 where we do pyruvate, pyrimidine synthesis and purine synthesis, where we get ATP at the very end. Okay, so first starting off for cellular respiration. So going through glycolysis, we have glucose seen here, where we form glucose 6-phosphate, which is very important. And then we go through the whole process. We're going to go all the way down through all these to get to pyruvate. And then once we get to pyruvate, we're going to move on to the citric acid cycle. So we go through the pyruvate dehydrogenase complex, and that's going to kick off through these 3 enzymes. We're going to get a byproduct of CO2, which is going to be very important later on. And moving on to the pyruvate, through pyruvate dehydrogenase complex, we're going to go to the citric acid cycle. So we get pyruvate moving in, and then we're going to crank out as many hydrogens as we can from this process, and we're going to move those hydrogens with NAD and NADH. FADH2, we're going to move these on into the electron transport chain, which is inside the inner membrane of mitochondria. And then inside of that, we got the ATP synthase. So we're going to separate the hydrogens from the electrons. We're going to make a concentration gradient, and the only way they can get back in, positive and the negative charges connecting back to each other, is going to be through the ATP is through this enzyme right here. So we're only gonna be able to shuttle the hydrogens down, make an even bigger hydrogen concentration gradient, and then ATP synthase. We're gonna crank these hydrogens back in. We're gonna turn this protein here. It's gonna make a ton of ATP. So from that whole entire process, we get a net of about 36 to 38 ATP. And so for the second process, focusing on ribose and nucleotide synthesis, we're going to go back to glycolysis. So we take glucose, we go through hexokinase, and we get glucose 6-phosphate. So we're going to take glucose 6-phosphate and go over to the pentose phosphate pathway. So we're going to use an NADP and then convert it to NADPH, which gives us ribulose 5-phosphate. And then we're going to move that and convert it into PRPP. And then we're going to move from page 1 all the way to page 4 with PRPP. And we're going to kick it in here. So PRPP can go through a lot of series and steps. So phosphorybosamine, and then glycosinamide, ribonucleotide. And then we're going to go through several steps to eventually form inosinate. And then inosinate is going to come over. So first we're going to use it for purine synthesis. So inosinate is going to go one of 2 ways. So it can either go and form ATP, which is going to be associated with adenine, or it can go through this process here, which will form GTP, and it's associated with guanine. And then going back to PRPP, so going to CO2, CO2 is going to be very important for the pyrimidine synthesis. So we're going to go through a conversion of steps with CO2. We're going to go to carbamic acid. We're going to eventually form orotate, and orotate is going to be kicked over to here. So pyrimidine synthesis is vital for orotate because it's going to convert into so we're also going to go from orotate, and then we're going to convert it to uridylate. And so uridylate can go from one of three ways actually. So it can go on to form UTP, which is associated with uridine. You can go to CTP, associated with cytosine, or it can go on and form TTP, which is associated with thymine. So we have A, T, G, C, and U. So through all that, we get ATP formed at the very end. So there's two processes. We can either go through cellular respiration, or we can go through ribosine nucleotide synthesis. So from there to there, all the way over to there. So that's my presentation. Let me know if you have any questions, and take care.